So we were also able to collect some samples that came out of these pillars that you see in the middle of this dried environment. Pillars, at the tip of them, were full of these crystals. And if you pull them out, if you can see, there's this green, there's a little bit of pink right there. Took those samples back, and we're trying to analyze them now to see, is that life? What is it? How is it surviving on there? So when we get them back into the lab, we're trying to figure out what kind of organisms they are. Are they on the surface of the crystals or inside one of these fluid inclusions? Because again, this is that salt crystal, and there could be some trapped inside of these fluid inclusions. This is a colorimetric test that we do to figure out what kinds of salts are present. And I can also do some analysis on anything that we find that is alive to try to figure out how they're surviving in the desiccated environment. And to do that, we are using these labs. We have these wonderful labs that were renovated with money from the Keck Foundation with all kinds of fabulous colors to keep us, attent and loving our lab environment. And you can see here, this is a sample of these halophiles that I grow as a comparison. So this is my lab strain that I grow as comparison. It's that lovely pink color. We have a huge amount of instrumentation at Carnegie that can be used for doing these types of analyses. So this is me, hairstyle number 3, in front of an electron microscope, which allows us to get a more detailed look at the surface. We have other microscopes that can be used. This is my boss, Dr. Andrew Steele. Taking a look down the microscope, we can look for fluorescence, and we can actually stain microbes to count them. This is Francis McCubbin, a very brave man, our sole male postdoc in a lab full of women. We give him points, or at least, padding. He's working in front of a laser spectroscopy unit that can be used to take a look at all the atoms, the bonding patterns between atoms in a sample to help identify samples. Another instrument is found in Marilyn Fogel's lab, and it's that Cyphergen machine that can be used to determine how much protein's in a sample, what kind of protein is it, and those types of expression analyses. So that's an overview of what it is that I do. I absolutely love my job. I get to be a child and perpetually ask why. We get to take a look at the strangest life on Earth, take a look at it in terms of human disease, pure curiosity, life elsewhere in the universe, and just to really step back and kind of go, wow, the world is a very strange and very wonderful place. So I just want to take a moment to just thank all the collaborators that I work with and everybody in my lab that's all here in the back. And thank you for your attention.
Thank you very much, Adrienne. I'm sure she'll be delighted to take questions.
I mentioned at the beginning that there's some thought that some of these organisms might be able to last thousands of years.
Yeah.
How can you determine they'll last thousands of years?
So the reason this is pretty controversial is where they found these samples is actually down old mines. They've gone down mine shafts and they found salt deposits down there, which the geology around it suggests the geology surrounding it is 250 million years old. But the question is, they do have halophiles living inside of those salt crystals, but the controversy is when were they actually included inside of it? And that right now is still very, very controversial. So I think it's one of those situations where it's good for the scientific environment for somebody to put that out there, and then for everybody else to try to figure that out. So, yeah.
A comment and a question.
Yes.
There's at least 2 caves in the world that have life forms that live in a acidic environment of like pH 1.
Yeah.
One's in southern Mexico, one's in eastern Romania. I don't know if that's of interest to you. The other is the extremophiles. I see maybe something that Nick Lowe wrote in his cell book, but something about are they very old life forms? They've been around for a long time.
So the rooting the tree of life is a hard thing to do. So taking a look right now at molecular clocks, we can figure out that some of these extremophiles are actually more closely related to you and I than they are to bacteria. But as far as time scales and distances, that's very controversial. There are people on all sides of that camp right now and trying to figure that out. So I wish them well, but I'd suggest wearing padding to those meetings because it's pretty controversial at the moment.
Is there a relationship between the way these salt organisms repair themselves and people repair, or your skin or bone heals?
So what makes it different, I'll start with what makes it different, is that these cells, it turns out that a lot of cells that can survive in extreme environments, they don't carry one copy of their genetic material in each cell the way that we do. They have many, and the halophiles can have up to 25 copies at any given time inside their cell. It's like they're carrying duplicates just in case for the repair. So there's more pieces to use as template for repair. That's probably one of the reasons why our cells are so fragile and so prone to things like cancers. What is common is how they repair at the end of the day, the mechanisms that they're using. So if we can figure out how those DNA repair proteins work in the archaea, then we can start to figure out what the proteins may be doing in people, because we don't have that sort of backup with copy number.
So if they have all those copies, do they have some sort of majority voting saying, like they do with some computer systems?
You know, how they actually, there's been a lot of ideas thrown out there about exactly how do the multiple copies work, because it's amazing. You can absolutely shred the DNA into tiny, tiny pieces, and it will reassemble perfectly, without a flaw. And how that happens is absolutely incredible. So there's been a lot of ideas about actually the structure of what that looks like inside a cell. And they're still tossing around models about that, but it seems like having more copies you have, the easier it is to find a piece that doesn't have a break where you have a break and match them up.
The name that was given to them archaea means, of course, the oldest, the ancient, which maybe in the beginning they thought they were.
Yeah, they did.
Doesn't some of the facts that you are speaking about suggest that they have a longer background? For instance, that they have extra copies for repair. Well, that's possible accumulation or possible wisdom, experience, that it helps to do that. So there may be other characteristics that suggest that.
You know, what's so interesting when you look at the extremophiles is that cells do not keep anything inside that they don't need. It takes a lot of energy to replicate DNA. So you don't keep pieces you don't need just hanging around. And yet, in the extremophiles, like this halophile that I've been studying, they have a copy of every known DNA repair system in the bacteria and in the eukaryotes. They have all of them, and some novel ones. They carry along a lot of extra baggage. You know, it seems like the Boy Scouts, always be prepared. And we talk about, you know, at the end of the world, the only thing to be surviving is the cockroaches, well, and the archaea. So there seems to be some sort of a suggestion there that they've been around for a while for a reason. They're packing enough baggage to survive a lot of changes that organisms like us, because we don't have a lot of backups, we wouldn't survive.
Are there confirmed examples of halophiles from elsewhere in the universe, say on meteorites or moving samples or anything?
No, there aren't. There was a lot of controversy with the Allen Hills meteorite, and some of you had seen in the news. And I'm going to, if you really want to get into a good argument about that sometime, talk to Dr. Andrew Steele about that. He's my boss. He actually was the one who wrote the paper saying that that wasn't life. So it's been a good, healthy argument, because I think it helps people to determine what is life, how do you sample, what does a sterile sampling technique look like when you're talking about these meteorites. So yeah, no, nothing has ever been confirmed as being life on another planet. But you have to remember that Viking landing happened over 30 years ago. And they're still arguing about whether the Viking mission actually confirmed there was life or not. So I think the argument in and of itself brings us closer to actually finding answers to some of these things. And some of the technologies that they test on things like the Amaze expedition are going to be the things that eventually go to Mars. They're going to be testing mission instruments that are going on ExoMars that's heading out to Mars with the European mission in this next round of Amaze expedition. So I think we're getting closer all the time to being able to answer these things.